Hello, everyone. Thank you for joining our webinar today, Formulation Approaches to Improve Microbial Stability. This webinar will be hosted by Ed Collins, our Director of Product and Process Development, with nearly 35 years of experience in the industry, and Dr. Malcolm Valladares, our food scientist with a plethora of processing expertise. My name is Drew Wellnitz, and I will be moderating this webinar. Ed, I will now pass it on over to you, and you may begin your presentation.
Thank you, Drew. Good morning. Today, Malcolm and I will talk a little bit about formulation approaches and some things you can do to improve microbial stability. Just as a little background, who is the National Food Lab? We are part of the Eurofins network. We have a long history in product and process development, sensory evaluation, and consumer research. And of course, we couple that with some of the food safety and microbiology testing of the Eurofins team, as well as all the supplements, chemistry, and other analysis that we do. When we get into the National Food Lab's general capabilities, we do everything from pipeline innovation, think about what's new, how are we going to make it, how do you look at those concepts, how do you get consumer feedback. We have a full suite of consumer research, whether it's central location tests, home use tests, co-creation with the appropriate stimulus. Then we move into product development, a good range of technical expertise all across applied food science, a lot of problem solving, a lot of how do I fix this? How can I use these ingredients? What are new ways to approach our problem? And then, of course, product development and optimization is part of that in our pilot plant. On the sensory side, it's fully trained and objective panel, quantitative and qualitative descriptive analysis, and a lot of shelf life testing, looking at when products fail and how they fail and what changes may occur. On process development, we have a full pilot plant out in Lafayette, Colorado. Everything from process development and thermal processes in a retort through to process letters, scale up, co-packer trials, everything that it takes to commercialize a product. And then we also do data management, so specifications, how it all ties together at the end of the day. And now I'll turn it back over to Malcolm to start talking about the topics for today's presentation.
Thank you, Ed. For today's presentation, we'll be focusing on the R&D tools that we can leverage and what are the strategies that we can use to enhance the microbial safety of the food products that you formulate. Basically, we'll be touching on the regulatory classification with emphasis on what are those parameters, key parameters that are relevant for the microbial stability from a food safety perspective. And also we'll discuss a little bit about the potential mitigation strategies you can use in terms of formulation, hurdles technology, and antimicrobial ingredients, and how the combination of those can be used to enhance the microbial stability of the products that you are formulating. In terms of the food microbiome, I like to call it, you have the good, the bad, and the ugly case. And basically, you have the good microorganisms, understand those as the microorganisms that have an economical value that are used to drive fermentation processes, and also probiotic microorganisms fall in the same category. And then you have the bad microorganisms, which are those that can be associated most commonly with food spoilage. And then you have the ugly ones, which are the ones that can cause disease and can cause lawsuits and economic loss for your companies. So those are all intermingled. The microbiome of a food product is always a dynamic scenario. And how do we prevent and what strategies we use to control them and make sure that we have the right microorganisms growing when you have a cultured product, or you can prevent spoilage microorganisms by controlling the food or controlling those intrinsic and extrinsic parameters, and how do you prevent or eliminate those ugly microorganisms that can cause disease, known as pathogenic microorganisms. So the food loss problem is one of the biggest drivers for the innovation in the industry, but we also have something called the freshness paradox, which consumers more and more are demanding foods that are fresher, that have minimal processing, and also this type of products are more vulnerable to spoilage by a wider range of microorganisms, because when you remove some of the hurdles, things that were not allowed to grow can start now becoming more prevalent in those type of niches. The food burden can also be estimated to at least $1.3 billion globally, which in the US accounts for almost a quarter of the food losses due to spoilage. And it can be caused by different means. You have the microbial spoilage, which is going to be the focus of this conversation, which is caused by the microorganisms and the enzymes released by them. And you have things like chemical spoilage, things like browning, enzymatic browning, oxidation of lipids, things of that nature. And the last, but not least common, is physical spoilage, which is mostly related to moisture losses or moisture absorption, things becoming soggy or stale. And food safety and food spoilage, food spoilage prevention and food safety are interconnected, and it's crucial to reduce the impact of those, because also it's going to reduce the economic losses of the companies in the long term. So when you start thinking about food regulatory classification, it's important to understand where your product lands in the regulatory framework landscape. Based on the intrinsic components or intrinsic characteristics of your product, mainly pH and water activity, you can have products that have low water activity and high water activity. The threshold here for water activity, the magic number, the key number is 0.85, and this is because that below 0.85, you eliminate the risk for pathogenic growth. In terms of pH, you have products that are low pH and high pH, and the number here is 4.6 for the same reason. 4.6 is the number that will stop pathogenic growth and also spores growth. In terms of what the processes that you can apply, when you have these combinations of parameters and factors, meaning for the example of this infographic, you have a low pH or a low acid food, meaning that the pH is greater than 4.6, and you have a water activity that is higher than 0.85, your processing options for having a stable product is basically you can do a hot fill or you can have an aseptic product. If you want, on the other hand, have something that is perishable or refrigerated product, the example of juices and sauces, that is going to be a pasteurized product that is just refrigerated. So based on how you want to position your product in your market and what storage condition and distribution channels you want to use, you need to play with these parameters and the processing that you apply in order to make your product stable from the microbial standpoint of view. So how do you define what regulation applies to your product? And this is a really nice decision tree that walks you through the different characteristics of your product and how the different regulations from the CFR might apply. I just want to mention from the get-go that Title 21 CFR 1 Part 117 will apply to all of them. So GMPs apply for the whole spectrum of products. But if you go in through the different decision nodes of the decision tree, and you start with a product that has, for example, water activity that is equal or below to 0.85, you're talking about products that are water activity controlled, meaning dry products, for example, crackers, things that are not going to spoil because the water available for microbial growth is very low. When you go on the other side of the bracket and you start looking for things that have a water activity that is above 0.85, you start asking questions whether you want to be a shelf-stable product or not shelf-stable. For non-shelf stable, you have all the refrigerated and frozen foods, which don't require any additional processing. For shelf-stable food products, you start asking the question whether the pH is another factor. If you are below or above 4.6, and then you have the case for low-acid canned foods and products that are acidified. For products that are acidified, you have a little extra steps where you need to define whether the majority of your ingredients are naturally acidic, or if your product is constituted by at least 10% by weight of non-acidic products, so you can define whether you are a formulated acid product or an acidified product. And this is another decision tree that kind of helps you to walk that path and define whether you are in one or the other of the buckets. Both are acidic foods, but one is just naturally acidic and the other one is formulated acidified food. So the important thing to remember here is that for formulated acid food, less than 10% by weight of ingredients should be non-acidified, meaning that the addition of that proportion of ingredients will not drive the pH drastically. Okay. Next, please.
All right, so now we'll talk about some microbial growth risk factors and how we work to control those, right? So the pathogenic and spoilage microorganisms need special conditions, right? And the acronym that we're applying to this is called FATTOM, right? So it's what's the type of food, what's the acidity of the product, how long are we going to treat it, what temperature are we going to treat it at, is there oxygen available to support growth, and then what's the moisture content of those products and how will that impact everything. So if we look at pH and water activity, it's a very common relationship. I think a lot of us are familiar with this. As you move the pH, the yeast, the mold, the bacteria all prefer to grow in different ranges and can go down to different levels for where they'll grow. And the different products that fall in, whether it's fruit juices, as you know, tomato products are kind of the high end of the high acid food range. You get some vegetable juices, and then when you move up into the meat, vegetables, milk, and some of the other products, you're clearly in the low acid area. So another common control on the microbial side is water activity. And you can see on the graph to the right, as you look at mold growth, yeast growth, and bacteria, there's ever increasing water activities. Most bacteria are done growing at about 0.88, and the yeast and molds go to much lower levels and are of a concern in each of those situations. So as you look at the microbial growth, and on this chart, you'll see that we have both microorganisms and yeast, and we'll talk a little bit about each of those and the types of products they reach. Malcolm had mentioned 0.85, right? The key about 0.85 is that it covers all of our pathogenic microorganisms. So Staph aureus being one of the last ones we worry about as far as decreasing water activity. Then you start getting into some of the yeasts and molds that'll grow. And finally, you get all the way down to the osmophilic yeasts, which are the most tolerant of changes in water activity. So from the FATTOM key learnings, the water activity will help reduce these. It's a critical control parameter, and the interaction between water activity and pH is a combination. It's actually the only combination outlined in the code. So in this case, Specialty Bar Company came to us. They're shipping product to Japan, and the product was going moldy over time. So it would get, however long it took on the container ship to get there. When it would arrive, you could open the containers, open the packages, and they'd be covered in mold. So to start, we looked at what's going on in their facility. Are there places where we can maybe help them with sanitation, where they may be contaminating ingredients? Are there temperature controls? What do the raw materials look like? Are there specifications that might say, well, here's where a higher microbial load is okay? And then finally looking at the water activity of the different bars and ingredients that they were using. So we got samples back of those bars. We looked at all of the different microbial analysis. So the plate count was low. The yeast and molds were also well within specification, nice and low. And then when we did a specific test for osmophilic yeast, they were at 55,000 CFU, pretty high. We typed that growth. The method that was used at the time couldn't distinguish between the two different types of organisms. So the Bailey is a common issue with fermentation and sugar syrups, and a candida is more common and tends to make alcohol. So as we looked at the microbial risks, right, we've got the ingredients, we've got the production environment, the processing and storage that we've just gone through. no major pieces in the production environment. And then we look at the microorganisms of concern, and you can start to see that the ones that we're concerned about down the yeast and mold box are showing up in this product. So what do we do next? We take a look at their product line. Let's see where they sit. Let's see what type of water activity they have. As you can see, they're all, there's one that's nice and low at 0.632. Most of the bars are 0.68, 0.7, fairly high, certainly below the microbial threshold, but not well below the yeast and mold threshold. We took a look at specific ingredients, and you can see here that the brown rice syrup is way too high, right? 0.782. They also had dates and raisins. The interesting thing there is that, because they're using whole pieces of some of these fruits and nuts, the highest allowable yeast and mold from a specification standpoint was on the dates and raisins, where the incoming counts could be as high as 5,000 and still be considered okay. And then candida is a well-known issue with dried fruits. So when you start looking at it and you say, okay, they've got brown rice syrup in there, what are some of the options? And these are 3 products that are offered off the Nature Biofoods website. You can see here, as you increase the dextrose equivalents from 0.28, from 28 up to 60, you get a subsequent drop in the water activity of the product. Obviously down here where it's 0.6 to 0.72 is the area where you want to be. That'll get you down in the safe zone for the yeast and molds, right? So the study points to the water activity being an issue. We looked at mix times with our product because they have a lot of pieces, whole pieces. The uniformity of the water activity just within the bar and from bar to bar was not uniform. So we recommended that they go up in longer mix times. And we did compare bar products or similar bar products against this. Typically, they were less than 0.65. So clearly, you want to get down a little bit lower. The current brown rice syrup, obviously too high. We put the new brown rice syrup in. We helped them source new suppliers for dates and raisins that had a lower spec. And the product now, ships to Japan, no problem, no yeast and mold, still going strong and on the market. Okay.
Great. So now I'd like to talk a little bit about food antimicrobials and how we can use those to enhance microbial safety and stability of our foods. So beyond the typical artificial or synthetic sources of antimicrobials, such as propionate and benzoates, the trend in the market now is to go and use more natural sources of antimicrobials. There are different types of antimicrobials that have the connotation of natural. And you have animal sources that are like lysosine, chitosan, which can also be from a mushroom source. And you have plant antimicrobials such as spices, well-known essential oils that can also act as an antioxidant. Hops and different extracts from garlic and onions have known or have shown some antimicrobial activity. But the most commonly known are microbial sources. And these are commercially available in different forms from different companies. And we have things like natamycin, which is an antimycotic, niacin and other bacteriocins like small peptides, and fermentates, meaning metabolites of fermentation, and protected cultures, which are meant to outgrow the population of other microorganisms. And bacteriophage, which is probably less known, and one of the examples of this technology being applied is for daily needs when you actually have a virus. The bacteriophages are viruses that are specifically designed to tackle a microbial organism. In this case, it's designed for Listeria monocytogenes. So how this differ from the traditional preservatives or synthetic systems. So the artificial or more commonly traditionally known preservatives like sodium benzoate, sorbates, and nitrates The advantage that they offer is that they have a well-documented efficiency, effectiveness against a broad spectrum of microorganisms, and they have a very low use level, meaning that you have to use very low, and they don't contribute to any off flavors or sensory changes to the product. Some of the cons is that they come along with a consumer perception that are usually connotated as bad ingredients, and the effectiveness of these compounds is limited by the pH. There's specific pH where they are more effective because they are usually in a disassociated form, and they're easily associated form, and they're easily absorbed by the microorganism. And some companies or some grocery manufacturers, some grocery stores have a naughty list of things that they don't like to see on the label. And therefore, more and more companies are trying to move away from these traditional systems to more natural or clean label options. So in this new bucket, we have, like I mentioned, things like bacteriocins, concentrated buffer lactic acid, acetic acid, which is vinegar, herbal extracts and essential oils, and these come with the advantage that they have a nice, they look nice on the label. They usually can be labeled as just a natural extract, so you might even get away with the claim of no preservatives added in your product. So some of the cons of these type of ingredients that they might require reformulation, meaning that you need to validate that this new ingredient will integrate well in your formula and will have the effectiveness necessary to mitigate the risk. Usually they have a higher usage level, meaning that in order to achieve the same lethality, you need to add a lot more in comparison to a traditional artificial preservative. And they might have some undesirable functional effects, meaning that they can change the flavor, they can change the color of your product due to shifts in pH or concentrated aromatic and flavor compounds coming from the natural extract. So here are some examples of common clean label antimicrobials. And you can see the first one is concentrated acidic buffer. Basically, it's a vinegar that has been buffered, and the use level goes from either 0.5 to 2.5. And the antimicrobial spectrum is very broad in this case. It can control against lactic acid bacteria, yeast and mold, but the sensory impact goes that you will have some more sharp, sour notes in your product. The labeling is good because you only can you can label it as a vinegar or natural flavoring, but you need to be aware of how this is going to look in your label. You need to make sure that it's not going to look strange or out of place. If you're looking at products that are dairy based, it's okay to have a fermented dairy antimicrobial because it's not going to look out of place. But if you have mushroom extract, that's going to look a little odd, and people might start asking questions, why I have a mushroom extract in my yogurt, for example. Next, please. So what are the goals of this antimicrobial systems? Once you put them in your product, what are the ultimate goal? Basically, your clean label system or your natural antimicrobial system needs to be able to deliver an equivalent lethality compared to your traditional system. So if you're replacing one ingredient to another, you need to make sure that you are achieving the same kill. Basically, for pathogenic microorganisms, you need to be able to deliver at least a 5-log reduction, meaning that you're going to be able to kill 5 logarithms of the specific target microorganism. And this is a combination of your formula, your process, and your antimicrobial. So you need to look at your formulation as a whole. In terms of spoilage, what do you need to achieve with your antimicrobial, natural antimicrobial system? Basically, you need to inhibit and control the growth over the shelf life of your product. So your product needs to have needs to be robust enough to withstand the abuse over the shelf life of the product. Meaning, if you have a salad dressing that is going to be you're going to open it, you're going to keep it in the fridge for months, your product should be able to withstand that temperature abuse of going in and out of the fridge and throughout the whole shelf life for those 4 or 6 months. Next, please. So what are the challenges when you are formulating products using these natural antimicrobials? First of all, you need to start asking the question, is this does this ingredient has a GRAS status? Are there any toxicological effects? It's going to trigger any allergenic reactions? And of course, there's going to be a cost component. Usually these ingredients, since they have a natural connotation, they have a higher cost per unit produced. And what is the form that you're going to be using it? Is it going to be used as a natural ingredient? It's going to be from the natural source, meaning that you're going to have celery powder, or you're going to go for an isolated version of the compound coming from the natural antimicrobial, which might be more effective, but also defeats the purpose of having a clean label product. And what are the sensory effects? Are going to have an impact on how the product tastes? Is it going to be acceptable by the consumers? Is it going to change the flavor of my product too much that no longer tastes like the product is supposed to be? And then you have things about food component interactions. The more complex the food system you are actually trying to put these ingredients to work, the less effective they become. Things also that you need to keep in mind when you're formulating this product is what is the validation method that you're going to use? How are you going to check if indeed this component or this compound is being effective in my formulation? And there are different ways to do it. You can think about starting with in vitro and then you do in situ or in product testing, but you can check for the antimicrobial activity in broad solution, agar diffusion, via turbidity methods. There are different ways that you can achieve that and generate enough evidence that your system is controlling against those microorganisms of interest. And then one of the things that usually gets forgotten is what is the influence of these antimicrobial compounds in the gut health? Is it going to change the natural microflora of your gut? Is it going to cause imbalances and more problems than actually the things it's meant to solve? So in a nutshell, the considerations you need to keep in mind when you're formulating products with antimicrobial is what is the effectiveness and how are you going to apply it into your food product? What is the polarity? Is it soluble in water? Is it soluble in oil? And what are the target microorganisms you want to control? Second, like I mentioned, is the food complexity and how it's going to perform. The more complex your item, the more complex your formulation, the less effective your component or your antimicrobial will be. And what is the delivery system? So you need to start thinking about also like how you're going to deliver that antimicrobial to actually make it more effective. Like the example here is like microemulsions of eugenol, which is an essential oil, has shown promising effectiveness against E. coli and Listeria when you use it in a microemulsion in the products, because the microorganisms are in the water phase. So you need to be able to deliver into that water phase. And since we are using an oil, having that emulsification step there makes it more bioavailable, and therefore it's more effective against target microorganisms. So that's in a nutshell. So this is one case study I want to share with you, and this is focused on a company that came to us, a client that had a line of different salad dressings, vinaigrettes, and creamy dressings, and they were non-thermally processed products, and they wanted to remove gluconol delta lactone from the label because it looked odd, and they were trying to achieve a new market where that product, that ingredient was banned. So the approach we followed, the general approach, the general goal was basically to identify potential ingredients that we could use as an alternative and start sourcing them. Also, we did a small lit review to determine what the microorganisms of importance, what are the target microorganisms and levels that we need to use for the challenge study further down the line. And also we needed to evaluate the sensory impact of these ingredients in the formulation. So we make the products with the known amount recommended use levels from the suppliers, and we verify whether or not they were compatible with the flavor profile, with the label, before we started and send this for a challenge study. So the general approach we follow here in our R&D lab was basically we started with the initial formulation, we replicated the formula here in our bench, and we started spiking these antimicrobials to do a sensory evaluation, a quick sensory assessment to determine whether these products were compatible or not. In some cases, we found that some of these antimicrobials were actually triggering curdling, so things were falling out of solution. There was a lot of phase separation, color changes, so those were weed out early in the process. Then we did the selection based on the antimicrobials that had a good sensory performance. And after that, we had the advantage that one of the ingredient suppliers had a microbial growth model, and we used the information from the formulation and sensory levels that we determined from the antimicrobials, and we feed that information into the model so they could create customized blends and customized levels of the antimicrobials, so they perform better in the different SKUs. And then we did a small sensory confirmation. We remade the samples with the recommended levels, and after that, we sent samples for a microbial challenge study to determine the robustness of the formulas. In all the selected antimicrobials were efficient, but in the specific case for this rose wine vinaigrette, we screened at least 13 different components, and the winner here was number 3, because it had a very mild, just a slightly more tart flavor in part of the formulation. And it was the best performing in terms of the microbial kill, and that's what was the formulation that was used for the challenge study. Yes.
So now we'll talk about some combinations and different approaches to control products. Many of you are familiar with hurdle technology, right? It's a combination of preservative techniques that alone may not preserve the product, but in combination make it difficult or impossible for the microorganisms to grow. It can be temperature, processing or storage or freezing. Water activity and acidity we've talked a little bit about, and we'll go into in some more depth and combination. Oxygen, preservatives as Malcolm reviewed, and then some other novel techniques as people find them, right? So in this case, we're looking at an existing product, right? It's a drinkable medical food. It's a multi-use container, which adds new hurdles to how you want to process it, right? It's high protein, high levels of hydrolyzed gelatin, right? 17 grams in a 30 ml serving. It's supposed to be shelf stable, no refrigeration required. And if you look at a stylized ingredient line for this product, it's hydrolyzed collagen, water, acids, a few amino acids, which are specific to its use. They have sorbate, benzoate, and sucralose, right? So the client is now asking us for 2 new versions. One, they want to look at an alternate collagen supplier that has a cleaner taste. And second, they want to look at, can we make a product with PDCAS of 1, and what level of protein, right, can we get into that 30 ml serving? So where do we start? What do we need to do? Took a look at the product. The current pH was 4.4. That's really in that borderline area here, where you can see that some pathogens will grow. Now remember, tomato paste and tomato products are really the only things that go up to about 4.6. If you look at a lot of thermally processed beverages, people like to be obviously less than 4.2. Some companies will not sell anything over 3.9. Just with that safety factor in measuring pH, having a little buffer is always a good idea. It's basically water, right? So the water activity is 0.97. And so you have to ask the question, are the current preservative levels sufficient to prevent the incidental contamination, right? So benzoate and sorbate tend to be better against yeast and molds. And then the secondary question is, would it be okay with a thermal process? Would that make this product safe, right? So you can clearly get it into the bottle with a thermal process, it'll be safe. And then because it's in the bottom right-hand side of the chart, above 4.2 with a very high water activity, there is the risk that you could potentially recontaminate the product, which means you're going to have to do some sort of challenge testing, right? So the water activity is too high. How can we improve the safety of the product, right? Get it down less than 0.85. then how do you measure whether that secondary incidental contamination will allow yeast and mold growth, right? Because that's where it'll start. You can change the pH, you can change the water activity when they start growing, and allow for the possibility of pathogen growth. So that's where the challenge study comes in, right? So you have to, as Malcolm did previously, you're going to have to measure that. You will have to contaminate the product and make sure there is some log reduction over time. And then what are the other product quality concerns that will come with it, right? And taste is going to be a big one whenever you look at water activity control, right? There's different ways to do it. We used the brown rice syrup earlier at 60 DE. That's great. That's not necessarily going to do it in this product. Glycerin, very good humectant, helps you drop the water activity very quickly. It's also bitter, right? So as you look at that change in water activity, the other thing that's going to change is how well you can hydrate the protein, right? So if we're looking at a PD-Cas1, what type of proteins are you going to use at this low pH? Well, it's going to be acidified whey. There's an additional bitterness that will come with that. There's obviously bitterness with the hydrolyzed collagen as well. And then whey protein, as we know, once you get above a certain concentration, you then have to address whether the product's going to gel or not. So the final solution, right? Obviously, we're looking at lowering the water activity. The one with dairy proteins became cloudy, right? Competition for water. You can get basically dehydration of the proteins as the glycerin will draw it out of the protein. We weren't able to get all the way to 17 grams with the products they wanted to use, right? So protein levels came down. And then secondarily, as you look at collagen, you have different levels of hydrolysates, right? So you have a 3,000 and a 5,000 Dalton, the 3,000 being more hydrolyzed, right? Smaller portions of the protein. Of course, that comes with a little more flavor as well. It also helps lower the viscosity, which is a bonus for the thicker products. How to use some flavor systems and some blockers. We also played with the different acid types, right? So are you using citric? Are you using lactic? Are you using malic? Are there other fruit acids that might accentuate or improve the flavor of the product? Finally getting down the 3.9 pH, right? From there, you look at it and say, all right, will this pass the microbial challenge test? Will it handle incidental contamination? We're now at 3.9 under 0.85 with still potassium sorbate and sodium benzoate in the solution. The product is shelf stable.
Right. Thank you, Ed. So kind of like to summarize and wrap it up this section, some of the things you need to consider when you're assessing the safety and shelf life of your product. So for the safety point of view, in terms of pathogenic microorganisms, what do you need to do, right? For your formulas and your products, you basically need to have PCQI or process authority to review your formula and process. They will be able to determine whether your food safety plan or HACCP plan is enough to control and mitigate the risk that you are introducing by your ingredients or your different processing steps. And also the process authority will be able to determine whether you need to file your process with the FDA and whether you need a processing authority letter or a scheduled process, basically. And determine also if you need a microbial challenge study to validate and provide enough sufficient data to the FDA to demonstrate that your product is safe by achieving enough lethality. So for this reason, you can use pathogen challenge studies to determine that you are indeed achieving that 5 log reduction under the storage conditions or processing conditions. And this, like I said, this data can be used to support a food safety plan and have that information submitted to the FDA. In terms of shelf life assessment for spoilage microorganisms, slightly different. You need to be able to determine the effectiveness of your system to inhibit the microorganisms that are causing the spoilage issue. And that needs to be done over the shelf life of your product, the desired shelf life of your product. And you need to consider the storing conditions For example, if you are going to have product shipped to Arizona, it's going to sit in the warehouse really in the middle of summer when it's really, really hot. Those are going to be extreme conditions that need to be accounted for when you're designing your challenge study. So whether you're going to distribute your products in the East Coast, West Coast, you need to consider all those different factors to determine the robustness of your system, to control also post-opening spoilage if you have a single-use, a multi-use type of presentation container. So those are factors that need to be taken into account. And usually it's recommended to run your shelf life at least 1.5 times the desired shelf life of your product, using the relevant conditions for storage. So this can be a measurement of the robustness after opening, and you need to keep in mind that also predictions will not be able to be made after or past the duration of the study. If you want to extend the shelf life of your product, you need to have a longer shelf life study. Otherwise, you cannot make conclusions out of it. In terms of the spoilage microbial studies or even pathogen microbial challenge studies, there are different ways to do it. You can have it by doing a broad screening, model system, and full-blown challenge study. And there are different pros and cons. So when you start talking about broad screening, this is just a quick, rapid method to assess the efficiency of your antimicrobial system in a broad scenario. So you basically spike the samples, spike the known amounts of your target microorganisms into a broth, and you apply your antimicrobial. This is the best case scenario because you won't have any interference from your product matrix, but this does not, the con is this will not include those extra ingredients. You don't know how it's going to perform in a more realistic scenario. The advantage is it has a short execution time but doesn't account for secondary growth. Then if you move forward into a model system, this is just like a stripped down version of your food product when you have just the macronutrients levels right, and you start using this to also do a quick screening of how these antimicrobial compounds will behave in your model system. And it's also very fast, so it's just a short timeline, it's relatively more affordable, but also doesn't account for secondary growth and might require still a full-blown challenge study to support your data for your food safety plan for the FDA. So a full challenge study, this is done in the final product. So you need to have product that is being made using the same equipment that you use for commercial product production. And for shelf life studies, you need to run it at least for 1.5 times the product shelf life of the product. And like I said, predictions cannot be made. Pros is this can also support the food safety plans, and the con is that it takes a long time because you need to account for the shelf life of the product. You need to account for the time that will take to grow the microorganisms, and you probably will like to run this at least in duplicate or not in triplicate. So each approach has its pros and cons. Some of them are just more useful upfront in the early stages of development, but the selection will depend on what stage are you at and what information you want to collect out of your study. So in a nutshell, what are the key learnings of the different approaches you can take? First of all, you need to understand where your product will fall into this space of the regulatory framework, right? Next thing you need to know is what are the risks. You need to be able to identify clearly what are the microbial risks that you're going to be facing based on your different formulation, different incoming ingredients, and the process steps that you actually have. So once you have that identified, you can start thinking about how you can tackle those risks using your formulation approaches, either by reducing water activity or playing with the pH of your formula or incorporating different antimicrobial compounds. And also at the end, you need to be able to validate that your system is robust enough to withstand microbial abuse throughout the shelf life of the product. So in a nutshell, this is the approach that we follow here at the NFL, and this is how we approach this type of projects. Very often we have to intervene in different stages, but if you look at it as a whole, these are the logical steps you need to follow in order to ensure or improve the microbial safety and stability of your products through formulation. Thank you very much.
All right, Drew, back over to you for any questions.
Absolutely. Thank you, Ed, and thank you, Malcolm, for your presentation. We'll move into the short Q&A. Remember, you can absolutely submit questions through the Q&A or through the questions tab. And if we do not address your question live, we will reach out to you after today's session. So it looks like our first question is, in your bar example, it looks like the product has dried fruits and nuts. Does changing water activity change the bar quality when it's a high protein bar?
Yes, so you face a lot of the same kind of issues. So the high protein bar is actually probably more difficult in that the proteins at that water activity will tend to harden over time. So you need to be conscious as you bring it down, looking at what proteins you're using. There's a whole suite of products out there. Each supplier has different proteins that are made for these types of conditions. They will start to set and harden and compete for water, and you will see changes in texture. So in that case, your product will be safe, but you have to look at the sensory acceptability, and that can change over 2, 3, 4 months. It's a much longer process to review on some of the acceptability changes.
Great, thank you. Our next question is, you talked about antimicrobials and salad dressings and the need for a challenge study. Would the challenge study help you determine release criteria?
Yes, that's a good question. So at the end, if you have two different prongs to this. If you have a product that is thermally treated, in this you have a salad dressing that is also being pasteurized, you can make sure that your product also, before you start the thermal process, you need to make sure that your product has achieved the right pH. So you need to make sure that you reach equilibrium. In that case, once you reach equilibrium, you can apply your thermal treatment, and then you determine that what is the survival for your finished product release criteria. In the other case of the example I had, those were non-thermally treated salad dressings. And the challenge study here is basically to determine the time that you actually need before you can release those products. So the time is required to achieve the 5-log lethality, relying on the acid itself, or relying on pH alone. So how long it takes to for the acid and the vinaigrette, rose wine vinaigrette, to actually deliver a 5-log kill of Listeria and E. coli. So that is the challenge study will help you to determine that time through different time points where you start measuring recovery, different levels of microorganisms on a period of time. And based on that, you can say you can release the product after 1 week if you store it at this condition, or after 5 days if you store it at higher temperature, because you allow the acid to react faster with the other microorganisms. And if you recall, the mechanism of action of how acid works to kill microorganisms is by when it's in the associated form, the acid can penetrate the cell wall, and the microorganisms will start to it will disassociate inside the cell, and the microorganisms will start pumping out hydrogen ions at the expense of ATP, so they will die out of exhaustion. So when you have a higher temperature for storing, that will drive that reaction faster. So in your challenge study, we basically validate the timing that you require in order to achieve that lethality. Hope that answers the question.
Thank you. Our next question is, what considerations do you need to keep in mind when formulating cultured products to prevent spoilage?
Good. That's a good question. So cultured products are products that basically are fermented. You have products like sauerkraut, which are basically, you drive the fermentation by selecting the microorganisms through the use of salt. So you are lowering the water activity, and you're allowing just specific microorganisms to grow. So some of the things you need to do, when you need to consider when you're doing that type of vegetable fermentation, is you need to start with high quality materials. So your supply chain controls in terms of good quality starting material, also sanitation controls are going to be key to make sure that you are indeed growing only the things you want to grow, especially in products that are not pasteurized. So you will find, like in the case of fermented vegetables, you find them in two different aisles of the grocery store. The ones that are refrigerated most likely are they have live culture, so they're still alive. They haven't been pasteurized, and the ones you can find on the shelf, those have been thermally treated. So at the end, the fermentation has been stopped by heat, and so in that case, spoilage is not going to be a concern because they are basically sterile products. When you're talking about cultured dairy, similar scenario, in the way you control spoilage is by starting with a clean sheet, with a clean canvas. Basically, you pasteurize the milk or your base that you're going to be using for fermentation to eliminate the competition. So basically, you kill all the competition or competing microflora, and you inoculate the product with the desired microorganism that you want to grow. And that, like the bacterioprotectant cultures, can be used because the fermentation will release secondary metabolites, which some of them have antimicrobial characteristics, and that will help you to control spoilage. And there's a fine line between fermentation and spoilage, and it's all depending on what microorganisms at what time and what level. So,
Thank you. Our next question is, when should I be testing my products for microbial contamination? Should I test my ingredients, in-process products, or just the finished products?
Okay, another good question.
There's kind of two pieces to this one, right? So it depends what your end product is. Malcolm had talked earlier about, is it refrigerated? Is it frozen? Is it ready to eat? What type of product are you making, right? There are some things that have very controlled limits, like if you're getting spray dried materials in, or if you're getting, sugar, those types of things have very low loads. It's a completely different story when you have, say, produce or other fresh products that come in, and how you treat those. So it's important to know what those two things are. Now, if you're looking at beverages and juices and things like that, you want to understand how clean those products are coming in, because as we talk about a 5-log reduction, you certainly don't want to start over 5 logs, right? So when you're looking at pasteurized beverages, you will want to know how clean it is coming in, to make sure that your thermal treatment's sufficient to have a shelf stable or a stable refrigerated product that's safe to eat. So you got to look at both the end use of the product and what those raw materials will be, right? So ready to cook, ready to eat, they'll all have different limits coming out of the manufacturing environment.
Thank you. Based on our timing, I'll ask just a couple more questions here. Our next question is, you had a couple pictures of salad dressings classified as acidified and acid foods. Can you please elaborate?
Right, that's a good question. Some of the, if you find products that have particulates, think about your creamy ranch dressing with blue cheese parts. The blue cheese chunks are actually not.
Creamy ranch dressing, blue cheese?
I'm mixing it up, sorry.
Blue cheese.
In this case, it's not potato, not tomato. Let's go with the blue cheese dressing. So the chunks of the blue cheese dressing are not naturally acidic. So in this case, those account for the less than 10% of your formulation being a low acid food. So therefore, that is a formulated acid food. But when you're talking about vinaigrettes are very simple. They only have your vinegar component and your oil component and some spices. In that case, the majority of the product is naturally acidic, so that counts as an acid food.
Thank you. And our next question is, do you need a pathogen challenge study for a product release if it has been classified as an acid food? Is the classification of a salad dressing related with a challenge study?
Let me think. No, the classification will be based on the intrinsic formulation factors itself. So whether it's not whether you meet that rule of having less than 10% by weight, and the challenge study will be determined whether you want to have a non-thermally treated process. You have a challenge study to demonstrate that your formula is enough or is capable enough to deliver the lethality needed for the product, the 5-log kill. If you need a process authority or you need to file with the FDA, it's because you're doing that using a thermal treatment. So your process authority will design the thermal treatment, and you don't need to rely on acid to release your product. Your product will be sterile at the end.
All right, and I'll just squeeze a couple more in here. Aside from water activity, what other factors should be considered in spray-dried protein powder powder products? Powder would be mixed with water by the consumer.
I think that's, do you want to?
No, please.
I mean, it's, so typically those are so low moisture that things don't tend to grow. Obviously, you want to keep those products stored in a low moisture environment, right? They'll pick up some moisture. They won't typically get liquid or get so high that something will grow. So any contamination would just come in as part of that original process. You can measure it, but growth after that point, until you have other nutrients and water, is going to be a minimal concern, right? You don't want to put your scoop back in your, Muscle Mass 3000 or whatever tub when it's wet, but the product's going to be safe after opening, as long as you're using something dry and keep it dry. Things don't tend to grow.
All right, and then our last question for the day is, will increased sugar content extend shelf life in beverages? Also, what do you think of agave nectar as a preservative?
So the agave, right?
Agave.
So the concentrations you need for sugars are really beyond what you would use in a beverage, right? So in like a ready to drink format. So agave, I mean, it's obviously shelf stable. I have it in oddly, I have it in my closet at home, right? So, those types of things, they have a very high sugar concentration. The water activity is low. Some of them tend to be a little bit acidic as well, right? Depending on how they're made, or something natural like that. Those will be shelf stable. In a beverage format, what are juices? 10, 12 bricks maybe. Those aren't high enough to have antimicrobial properties. It tends to be the acid in those systems is more conducive to microbial stability than the sugar is. So you wouldn't go high enough where sugar would change the shelf life of a beverage. It would be more in thermal processing and pH.
All right, thank you very much. Based on our timing today, that will wrap up our Q&A portion. And thank you, Ed, and thank you, Malcolm, for your time, your expertise, and your presentation today. Thank you, everyone.
Thanks, everybody.